Okay, let's unpack this. Usually when we talk about fundamental biological drives, we operate into this assumption of simple math.
Right, the basic input-output equation.
Exactly. We treat the body like an engine running out of gas. A car drives until the tank is empty, and then it just docks.
It is a comforting way to view physiology, honestly. We really prefer that straightforward idea where physical depletion equals a full behavioral stop.
Because it makes sense. You either have the fuel to keep going, or you do not. It feels very clean and binary.
The problem is that biology, well, it rarely cooperates with our preference for simplicity.
Which becomes glaringly obvious the second you step into the world of behavioral neuroendocrinology.
Oh, absolutely.
Because in that specific field, you find that an empty gas tank can magically refill itself simply because you, painted the car a different color.
That is a great way to put it. It completely defies the simple math.
It really does. We're looking at a landscape of motivation and biology that is incredibly murky. And that is exactly our mission for you today.
We have a lot of ground to cover.
We do. We are taking a deep dive into a comprehensive 2026 research report. It's titled The Coolidge Effect, a deep dive into sexual novelty, habituation, dopamine, evolution, and human relationships.
It is a massive piece of research.
And our goal here is to cut straight through the folklore, the internet myths, and, frankly, the very loud voices of biological determinism.
Yes, there is so much misinformation out there.
So much. We want to understand the actual verified science of sexual novelty, how habituation mechanically works, and what it really means for long-term human relationships.
Because the cultural narrative surrounding this topic has wildly outpaced the scientific reality, which makes this exploration critical.
I mean, even the name itself is fascinating. The Coolidge Effect.
The origin story is completely wild, though historically quite dubious.
Right. It involves the 30th U.S. President, Calvin Coolidge.
And First Lady Grace Coolidge.
Yes. The story goes that they were touring a farm, but they were touring it separately.
And the First Lady is shown a rooster mating enthusiastically, many times a day.
Just going at it.
Right. And she supposedly tells the farmer, point that out to the president when he comes by.
I always picture the farmer sweating nervously in this scenario, just caught between the president and the First Lady.
Oh, it puts the farmer in a rather precarious diplomatic position for sure.
Definitely.
So the president comes along, the farmer nervously relays the message, and Coolidge asks, same hen every time.
And the farmer says...
His farmer says, oh no, Mr. President, a different hen every time. And the president replies, tell that to Mrs. Coolidge.
It is the ultimate historical mic drop.
It really is.
Even if it probably never happened exactly that way. But the term stuck. The report notes that Frank A. Beach started using the term in the late 1950s.
Yes. Beach was a prominent behavioral endocrinologist, and what's fascinating here is that the survival of that joke and its transition into official scientific nomenclature, it isn't just a matter of scientists appreciating a punchline.
Right, it wasn't just an inside joke.
No, it survived because it perfectly encapsulates a genuine, incredibly frustrating experimental problem in the laboratory.
How so?
For decades, researchers struggled to distinguish between two completely different physiological states. Absolute physical generalized exhaustion versus a partner-specific motivational decline.
Because if an animal stops a behavior, you don't instantly know why it stopped.
Exactly. You just see an animal lying down.
You just see a tired rat. But to figure out if this effect is real, we have to establish the baseline. We have to look at how scientists originally proved this wasn't just a case of an animal being too physically tired to continue.
The foundation of this comes from Beach and Jordan's 1956 work on male rats. They really needed to establish a rigorous baseline of, well, sexual exhaustion.
What does that actually mean, though? What is the strict criterion in a 1950s laboratory setting? How do you scientifically prove a rat is absolutely exhausted?
They took 12 male rats and permitted them to copulate repeatedly with receptive females until they reached this strict criterion. They monitored the animals for a continuous absence of sexual behavior over a very specific time frame.
Okay, so they timed it.
Yes. They waited for the male to completely stop responding to the female for a full 30 minutes. They didn't just observe a quick pause. They observed a prolonged behavioral shutdown.
Wow, 30 minutes is a long time for a rat.
It is. They were then retested after varying recovery intervals, 24 hours, a week, to see how long the physical systems actually took to reset.
Which proves that repeated sexual performance has very real, measurable physiological limits.
Precisely. The animal eventually just stops.
So they established the baseline. The rat is definitively tapped out. But how do you prove it isn't just a tired muscle?
You introduce decisive control. You change the stimulus.
Ah,
Imagine a male rat that has completely stopped mounting a receptive female and has been unresponsive for that full 30-minute window. He is, by all observable metrics, exhausted. If generalized physical muscle fatigue or absolute physiological incapacity were the entire explanation, swapping out the female should do absolutely nothing.
because a tired muscle is a tired muscle, regardless of what is standing in front of it.
Exactly. But when the researchers introduced an unfamiliar, novel, receptive female to that supposedly exhausted male, he resumed sexual behavior almost immediately.
That is wild.
The behavioral shutdown simply could not be explained by muscle fatigue. The animal acts as its own control here.
So you have a familiar partner first, leading to a complete shutdown.
Yes, and a novel partner second, leading to an immediate restart, with almost zero time for actual physiological recovery in between.
It's like sensory-specific satiety.
Yeah.
Like when you are sitting at the dinner table at Thanksgiving and you have eaten so much that you are physically uncomfortable,
Right, you cannot take another bite.
you are entirely too full for even one more single bite of turkey, but then someone brings out a completely new, visually stunning pie, and suddenly you have room.
It is a very similar mechanism.
The physiological state of your stomach capacity hasn't changed in those 5 minutes, but the novel stimulus has completely overridden the satiety signal.
The underlying architecture of habituation there is strikingly similar, yes. However, we do need to clarify a crucial biological limit here.
Okay, what's the limit?
Because this is a primary area where public understanding of the Coolidge effect really diverges from reality. Novelty resets motivation within strict constraints. It does not grant an organism infinite physical capacity.
That feels like a really important caveat.
Yeah.
Because the whole rooster story implies he just goes forever, all day, with limitless stamina.
And the research vehemently contradicts the idea of limitless stamina.
It does.
Absolutely. Repeated access to novel partners can certainly extend sexual responding far beyond what happens with a single partner. But male animals eventually reach deeper, absolute forms of sexual satiety and physical exhaustion.
So they do actually run out of gas eventually.
They do. Even if the motor patterns continue, meaning the animal is highly motivated by the novelty to continue mounting, the actual seminal output, the ejaculation, eventually ceases entirely.
Oh, wow.
The physiological machinery taps out, even if the brain is still demanding action. It is a motivational reset, not a magic spell for infinite capacity.
Okay, so the body still has hard limits, but the threshold to reach those limits is pushed further back by novelty.
Precisely.
And the report highlights that this is a reality across a massive swath of the animal kingdom. We are talking mammals, birds, fish, insects, even simultaneous hermaphrodites.
It is incredibly widespread.
I have to admit, that completely blew my mind. How does a hermaphrodite even show the Coolidge effect? What does that even look like?
It is a fascinating evolutionary adaptation. Let us take certain species of pond snails as an example.
Okay, pond snails?
They are simultaneous hermaphrodites, meaning they possess both male and female reproductive organs at the same time. Now, sperm production takes massive metabolic energy. It is a finite and costly resource.
Right, they can't just waste it.
Exactly. When they encounter a novel partner, they do not just increase mating frequency, they literally alter their sperm allocation.
Wait, really? They calculate how much sperm to use based on whether they recognize the snail.
In essence, yes. If they are mating with a familiar partner, they conserve their sperm, allocating a much smaller volume because they have already fertilized that specific genetic line.
The biological return on investment is diminishing.
Correct. But when presented with a novel partner, a completely new genetic opportunity, they significantly increase the volume of sperm transferred.
That is so strategic.
It is. And the fact that this cross-species diversity exists suggests that novelty influences several different components of reproductive investment, not just the raw frequency of copulation.
If it's this widespread across all these different types of species, it brings up a huge question for me. The internet often claims this is strictly a male phenomena. Is that what the animal data actually says? Are female animals just completely immune to this novelty effect?
The animal data firmly corrects that misconception. Female animals do show novelty responses, but the historical research on this has been fundamentally skewed.
Skewed how?
For a long time, the scientific endpoints were heavily biased toward male physiology. Researchers were looking for ejaculation-based measures or the sheer number of mounts, which obviously apply to males.
Right. If your entire criteria for measuring sexual motivation is based on a physiological event that females literally don't experience in the same way, you're going to conclude they don't have the motivation.
Exactly. It is a measurement error. But let us look at the 2018 hormonal review by Ventura Aquino and Fernandez Guasti, and even going back to Lisk's observational data in 1982.
What did they find?
When researchers actually look for it using appropriate metrics, female golden hamsters and rats absolutely demonstrate Coolidge-like effects.
How do they measure it then if they aren't using those male-centric physical endpoints?
They look at something called perceptive motivation.
I definitely need a translation for perceptive motivation.
Think of it as the active, solicitous behaviors in animal displays to invite mating. It is not just receptive, which is simply allowing mating to happen. It is perceptive, meaning actively initiating it.
Oh, okay. So they're seeking it out.
Yes. Researchers measure renewed acceptance of a new partner, altered pacing behaviors, and how the female regulates the mating location when a new partner is introduced.
What does pacing behavior mean in this context?
For example, a female rat in a specialized testing chamber will control how often the male can access her by darting away and then returning. When a novel male is introduced, she alters that pacing to allow more frequent access.
Oh, I see.
The female endpoints are highly motivational and context-dependent. The issue was never that females lack the mechanism. It is that the behavior is less uniform and relies on completely different metrics than the male model.
Okay, so the behavior happens in males, females, snails, hamsters across the board. But how does a new partner magically erase that fatigue?
That is the million-dollar question.
Right. If we want to understand what this means for complex human beings, we have to look under the hood at the brain's circuitry.
We do. And the engine driving this phenomenon is the mesolimbic motivational system. Specifically, we are looking at the dopamine pathways that project from a region called the ventral tegmental area, or VTA, to the nucleus accumbens.
Can we break those two areas down, the VTA and the nucleus accumbens? What are their actual jobs in this scenario?
You can conceptualize the VTA as a sort of deep brain sensor and engine room. It constantly monitors the environment for biologically relevant stimuli, food, danger, a potential mate.
Okay.
When it detects something important, it manufactures dopamine and fires it forward into the nucleus accumbens. And the nucleus accumbens acts as the action center, or the steering wheel.
So the VTA is the engine and the nucleus accumbens is the steering wheel.
Exactly. When it receives that dopamine, it translates that chemical signal into physical motivated action. It tells the organism, move toward that thing.
To see this engine room and steering wheel in action, the report highlights one of the most famous pieces of direct evidence in this field, the 1997 microdialysis experiment by Fiorino, Curry, and Phillips.
An incredibly elegant study.
And the methodology here is incredible because they didn't just watch the rats from the outside, they literally tracked the dopamine in the nucleus accumbens in real time.
The technique of microdialysis involves inserting a microscopic probe, a cannula, into a specific millimeter-wide region of a living rat's brain.
Wait, while the rat is awake?
Yes, the animal is awake and moving freely. This probe collects minute samples of the extracellular fluid in the brain every few minutes, giving us a real-time chemical readout of what the brain is doing during specific behaviors.
That is amazing. So they monitor the brain chemistry during initial copulation, during the onset of satiety, during the reintroduction of the exact same female, and finally, during the introduction of a novel female.
Yes, those were the 4 distinct phases.
What did that readout actually look like?
The data from that probe provided a stark, undeniable picture. When the rat began initial sexual activity, the extracellular dopamine levels in the nucleus accumbens spiked dramatically.
Just a huge surge.
It was a massive surge of neurochemical activity, but as the animal populated over time and eventually became sexually satiated, its physical behavior declined. And simultaneously, the dopamine activity plummeted right back down to the baseline resting level.
The engine room basically stopped sending fuel to the steering wheel.
Exactly.
But here's the critical test. When they took the familiar female away and then put her back in.
Nothing. The dopamine levels stayed completely flat. There was no motivational rebound whatsoever.
Wow.
The brain recognized the stimulus as already processed and deemed it unworthy of further neurochemical investment.
But the moment the novel, unfamiliar female was introduced into the chamber,
the dopamine spiked again instantly, followed immediately by renewed copulation, which drove the dopamine even higher.
that is just incredible to actually see on a graph.
It is an incredibly powerful piece of mechanistic evidence because you are watching the invisible neurochemical change happen across the exact moment of behavioral transition.
But this is where we have to pause and do some serious myth busting. Because when anyone hears the phrase dopamine spike, they instantly translate that to pleasure.
Yes, that is a very common misunderstanding.
The assumption is that the new rat is providing more pleasure than the old rat, and therefore the dopamine is higher.
And this raises an important question regarding how we view brain chemicals. The report explicitly dismantles that assumption, and it requires us to fundamentally redefine dopamine in pop culture.
Right.
In contemporary neuroscience, dopamine is not a direct meter of subjective enjoyment. It is not the pleasure chemical.
If it isn't pleasure, what is it? I mean, if you eat a piece of incredible chocolate, isn't that dopamine making it taste good?
Actually, no. The actual subjective pleasure you feel, the liking of the taste of the chocolate upon your tongue, is governed by entirely different systems, primarily the opioid and endocannabinoid systems. Yes, dopamine operates before the chocolate is even in your mouth. Dopamine is about incentive salience. It is about learning, prediction, vigor, and motivated approach. It is the chemical of wanting, not liking.
So dopamine isn't the reward itself. It's the brain's highlighter pen.
I love that analogy.
It highlights a stimulus in the environment and says, pay attention to this is relevant, this is unresolved, go get it. It creates the drive to pursue, not the satisfaction of the capture.
The highlighter pen analogy captures the function perfectly. A novel partner has incredibly high informational and incentive value precisely because it differs from the internal representation of the recently encountered partner.
It's new information.
Exactly. The brain highlights the novel stimulus because it is an unresolved variable. It requires investigation. The dopamine surge is the brain demanding that investigation.
But reducing complex dynamic mating behavior down to just one neurotransmitter, just dopamine, feels dangerously simplistic. I mean, are we really just biological puppets on a single dopamine string?
The researchers in the report entirely reject that reductionist view. The strongest neurobiological evidence actually supports what is termed a network account.
A network account, okay.
Dopamine is prominent. Yes, it is the initiator, but it is entirely insufficient as a complete explanation. When an animal reaches sexual satiety, it isn't just that dopamine drops. The body produces massive cascading changes in steroid hormone signaling and multiple other neurotransmitter systems.
What else is happening in that network? If dopamine is the gas pedal, what are the brakes?
You have a massive influx of serotonin, endogenous opioids, and prolactin.
Prolactin.
Yes, prolactin in particular acts as a profound refractory signaling hormone. These chemicals physically bind to receptors that dampen arousal. They actively influence sexual inhibition.
So it's an active stop signal, not just a lack of go.
Precisely. It takes this massive, coordinated cocktail of serotonin, opioids, and prolactin to put a biochemical lock on the physical arousal system and tell the body to stop. And it takes a surge of dopamine to override that lock and kickstart the system again. It is a highly complex, multivariable battle of neurochemistry.
And there is another layer to this that I found absolutely fascinating in the report. For novelty to even exist as a concept in the brain, memory has to exist.
Yes, that is a crucial point.
You cannot be surprised by something new if you don't remember what is old.
Novelty is not an intrinsic physical property of an object or a person. It is a relational property based entirely on the observer's memory. The report cites a brilliant 2003 study by Petroulis and Eichenbaum on golden hamsters to prove this.
What do they do?
They looked at a specific part of the brain called the peripheral entorhinal cortex.
Which does what exactly?
It handles individual recognition memory. It is the specific neural circuitry that allows an organism to catalog a scent or a face and say, I know you, we have met.
What happens if you take that circuitry offline?
When the researchers disrupted that specific memory circuitry, the hamsters lost their normal preference for investigating a novel female. The Coolidge effect completely disappeared.
Because they couldn't remember the old partner.
Precisely. Without the memory of the familiar partner stored and accessible, every single interaction felt entirely novel, or, conversely, the novel partner could not be distinguished from the familiar one.
That makes total sense.
You simply cannot respond to novelty if your brain cannot verify who is familiar. In rodents, this individual recognition is heavily reliant on olfactory cues. They memorize the intricate scent profile of the partner's pheromones.
Which brings us to the most complex transition of this entire deep dive. Because human beings are not golden hamsters relying on a pheromone profile to identify our partners.
We certainly are not.
We have language, We have incredibly complex long-term memory. We have culture. We make vows. We have ethical frameworks. So how does this deep brain circuitry, this ancient habituation and dishabituation, this dopamine highlighting system, operate in humans?
Translating this neurobiology to humans is where the science faces immense ethical and practical hurdles.
I can imagine.
We cannot ethically replicate the Fiorino dopamine study with humans. We cannot take a human subject into a laboratory, instruct them to engage in sexual activity to absolute satiety with their spouse, and then casually introduce a novel, receptive human partner to see what their nucleus accumbens does.
Right. I think we're all very grateful for those ethical review boards.
It is ethically prohibited and practically absurd.
But if we can't run the exact experiment, how do we know the human brain works the same way?
Human research is forced to rely on proxies. We use laboratory stand-ins for the actual experience of partner novelty.
Like what?
This includes repeated exposure to erotic films or images. We measure psychophysiology like blood flow to the genitals, or we track subjective arousal ratings, or we use attentional measures like sophisticated eye-tracking software. We utilize habituation paradigms.
And the report highlights the work of Kukunas and Stone. Over on this specific paradigm, they brought men into the lab and repeatedly exposed them to the exact same erotic video material.
Yes.
And they found exactly what you'd expect. Clear, measurable decreases in sexual arousal and attentional engagement across repetitions. The men habituated.
Their brains categorized the video as known information and dialed down the response.
But when the stimulus conditions changed, a new video, new actors, the arousal recovered immediately.
Which demonstrates the underlying neural architecture. Human males clearly show habituation and dishabituation to sexual stimuli.
Okay.
However, we must heavily qualify this data. The novel partner in these proxy studies is represented on a two-dimensional screen. It is an image lacking social context, smell, touch, and relational dynamics. It is an imperfect proxy for a real, breathing mating partner.
The use of visual proxies is really interesting, though, because for decades, the scientific consensus was that women didn't show these same habituation and dishabituation patterns to visual stimuli.
That was the prevailing theory for a long time.
The narrative was that women's arousal was purely emotional or relational, not triggered by visual novelty. But here's where it gets really interesting. We get to the pivotal 2013 study by Dawson, Sushinsky, and Lumiere, where they compared men and women directly.
That 2013 study is vital because it forcefully challenges those long-standing cultural and scientific assumptions. They didn't rely on questionnaires. They measured genital response patterns directly.
Right, getting the actual physical data.
Yes. Using physiological sensors, plethysmography for men, and vaginal photoplethysmography or thermal imaging for women, they monitored actual blood flow to the genitals during repeated exposure to stimuli.
And what did the physical data show?
It showed that men and women displayed remarkably similar physiological patterns.
Wow.
Both sexes showed clear, undeniable habituation to the familiar erotic stimuli. The physical arousal dropped. And crucially, both sexes showed clear dishabituation, a rapid physiological recovery, when the novel stimulus was introduced.
So the core physiological hardware for detecting and responding to sexual novelty exists and operates strongly in both men and women.
Absolutely.
So why on earth did we think for so long that women didn't have this physical response? If the hardware is there, how did scientists miss it for decades? The report mentions earlier flawed studies, like the 1995 work by Lauren and Everard.
It comes down to experimental methodology and a statistical problem known as floor effects.
Can you explain a floor effect?
In some of those earlier studies from the 1990s, the erotic slides or films they selected to show the female subjects were simply too weak, too clinical, or uninteresting to elicit a strong initial genital response.
Ah, I see.
If you show someone something they do not find highly arousing on the first viewing, their physiological response starts near zero. If you show it to them 10 times in a row, the response stays near zero. You cannot scientifically measure a decline at habituation if the response is already resting on the floor.
That's like trying to prove my car's brakes work while it's parked. If the speedometer already says 0, you can't prove the car is slowing down. You have to get the car moving at 60 miles an hour first before you can measure the deceleration.
A perfect analogy. When later researchers like Dawson utilized more effective, highly arousing stimuli and implemented better measurement intervals, the habituation pattern in women became blatantly clear.
The car was moving, so they could finally measure the braking.
Precisely. Furthermore, We must account for the fact that women's genital arousal often shows much lower concordance with their self-reported subjective arousal compared to men.
Meaning, the body is doing one thing, but the brain is reporting something else.
Exactly. A woman's body might be responding physically increasing blood flow to the visual novelty, but her conscious mind, when filling out the questionnaire, is reporting low subjective arousal based on context, perceived safety, cultural conditioning, or lack of emotional connection to the stimuli.
Oh, that is fascinating.
The physical reflex happens, but the psychological interpretation suppresses the reported desire.
But I want to push back on this because it feels like there's a massive contradiction here. If men and women show the exact same physiological habituation and dishabituation in the laboratory, why do massive global surveys consistently say that men want more partners?
That is a very valid question.
I mean, if the hardware is the same, why is the behavior so different out in the real world?
It appears contradictory until you separate two fundamentally different concepts. The report utilizes a massive 2003 survey by Schmidt and colleagues, which is a landmark study that included over 16,000 participants across 52 different nations and continents.
That is a huge sample size.
It is, and it pairs this with a 2021 experimental study by Hughes. These studies across diverse cultures consistently find that men, on average, report a much stronger desire for sexual variety and express a preference for greater variety among potential short-term mates.
So how do we reconcile 16,000 people saying men want more variety with the lab data saying both sexes react to variety the exact same way?
We draw a critical distinction between a reflex and a goal. A basic physiological response to novelty, the genital dishabituation, the raw blood flow we see in the lab, is completely different from a psychological preference or the active real-world pursuit of multiple partners.
Okay, I think I see what you're saying. It's like my salivary glands might react identically to the smell of a dozen different types of fast food restaurants if I walk past them on the street.
Yes.
That is my physiological response to novelty. I can't control my saliva,
Huh.
but my psychological preference, my health values, and my active choices might dictate that I only ever eat a home-cooked meal.
That is exactly it.
The physical hardware is shared, but the psychological software makes a completely different choice.
The software is shaped by evolution, culture, risk assessment, and context, and that is where the average sex differences emerge. However, the report is very careful to emphasize that we must be incredibly cautious with these population averages,
Why is that?
because they often obscure massive within-sex variation.
Meaning, not all men are the same, and not all women are the same.
Not even close. We are dealing with overlapping bell curves. The statistical average man might report wanting more variety than the statistical average woman. But when you look at the individual level, factors like sociosexuality, which is a person's willingness to engage in sexual activity outside of a committed relationship, play a massive role.
So, a woman with high sociosexuality might desire much more variety than a man with low sociosexuality.
Assuredly.
Yeah.
Add in cultural background, age, hormone status, and individual personality traits, and you create enormous variations.
Right.
You will routinely find that the difference in novelty-seeking behavior between two specific men is vastly larger than the difference between the average man and the average woman.
That makes a lot of sense.
The Coolidge effect is a population-level scientific concept. It describes a general mechanism. It is not a diagnostic label you can confidently slap on an individual to predict their personal relationship behavior.
So if this novelty detection system, this physiological hardware, this dopamine highlighting system exists across so many species, including both human men and women, evolutionary biology must have a reason for it.
It absolutely does.
Nature doesn't build complex neural circuitry by accident. What is the evolutionary why behind this?
To comprehend the evolutionary why, we have to examine the cold, hard, unromantic math of reproduction. Specifically, we must look at minimum obligatory reproductive investment.
We are talking about the biological cost of making a baby. Gestation is incredibly expensive.
It is an immense metabolic and temporal cost. In many mammals, including humans, a male's minimum obligatory physiological cost for reproduction is relatively low.
It's just the sperm.
It is the contribution of sperm. The biological commitment can be over in minutes. A female's obligatory cost, however, involves 9 months of internal gestation, immense caloric transfer, the physical risks and vulnerabilities of birth, and traditionally, years of lactation and direct physical care.
So if we look at this purely as a numbers game from an evolutionary perspective,
the math dictates that a male mammal can theoretically increase his total reproductive output, the sheer number of offspring carrying his genes into the next generation, simply by securing additional matings with new partners. His ceiling is high.
Right. There's practically no limit for
A
him.
female, conversely, cannot dramatically increase her total offspring number simply by mating with 20 different males in a single year. She can still only have one pregnancy at a time.
Right.
Therefore, under these ancestral conditions, a male motivational system that recovers quickly when new fertile partners become available is highly adaptive. It maximizes his genetic fitness.
The report actually cites an observational study by Jokela and colleagues from 2010 that looked at this dynamic in modern humans, which I found fascinating.
The Jokela study is very revealing.
They analyzed data from thousands of U.S. men and women over a long period, and they found that serial monogamy, moving from one committed relationship, having children, and then moving to another committed relationship over time, was positively associated with total reproductive success in men, but not in women. The guys who had multiple serial partners over their lifetime ended up with significantly more children overall, which seems to perfectly align with that evolutionary math you just described.
It is entirely consistent with the hypothesis of an evolved male novelty mechanism designed to maximize reproductive output. However, we must ensure we are presenting a balanced narrative here.
Right, we can't ignore the female side.
Exactly, because evolutionary biology is not solely focused on male strategies. Female novelty seeking also has strong, documented evolutionary drivers, but they are not based on raw volume. They are more heavily dependent on ecology, resource acquisition, and genetic quality.
But if gestation is so expensive for a female, wasting energy on a new mate makes zero sense to me. If she isn't increasing the quantity of her offspring, what is her biological return on investment for seeking novelty?
The return on investment for the female is quality, diversity, and survival probability. Mating with multiple or novel males can provide vital genetic diversity to her offspring, which acts as an evolutionary hedge against disease and parasites.
Oh, so a diverse brood is more likely to have survivors if a plague comes through.
Precisely. It can also help avoid sperm depletion if her primary mate is infertile or subfertile. Furthermore, in some primate and mammalian species, mating with multiple males actively confuses paternity.
Wait, why would a female want to confuse paternity?
Because it protects her infants from infanticide. If multiple males in a group believe an infant might potentially be theirs, they are far less likely to kill it and might even offer incidental protection.
That is so dark, but evolutionarily brilliant.
Additionally, novelty seeking can increase her access to resources, food, or territory provided by different males.
So the female evolutionary motivation for novelty is more like a strategic, highly contextual portfolio diversification, whereas the male motivation is more about raw volume and spreading the investment as widely as possible.
That is a very apt way to summarize the dominant evolutionary theories. And the variation in these female benefits across different species and environments helps explain why female Coolidge-like effects in the laboratory are sometimes less uniform than the male response.
Because it's contextual.
Yes. The female system is designed by evolution to be highly context-sensitive, assessing risk versus reward, rather than a simple on-off switch for frequency.
This all makes logical sense from a biological perspective. But I can hear people listening to this right now, especially people who are struggling with fidelity or boredom in their relationships, and thinking, well, the math is the math. Evolution built me this way.
That is a very common reaction.
They might think, I have this dopamine system highlighting novelty. Therefore, monogamy is unnatural, cheating is biologically inevitable, and I have a scientific excuse for my behavior. How do we respond to that kind of deterministic thinking?
If we connect this to the bigger picture, it is vital that we directly refute that thinking. It is a massive logical leap known as biological determinism, and it is fundamentally flawed.
Okay, let's break that down.
Yes, we have evolved capacities for sexual novelty. The dopamine pathways exist, but evolution is not destiny. A biological predisposition is not a behavioral command.
We aren't just walking dopamine receptors helplessly following a chemical trail.
Let me stop you there, because the data says something even more complex than simply we have free will. It is that humans possess multiple, simultaneously active, and often competing evolutionary systems.
Computing systems.
We have an evolved novelty detection system, yes, but we also have an incredibly powerful, equally evolved capacity for long-term pair bonding. We have profound neurobiological attachment systems driven by oxytocin and vasopressin.
Right.
We experience deep-seated evolutionary jealousy. We have advanced executive control in our prefrontal cortex that allows allows for complex long-term planning, impulse inhibition, and delayed gratification. We have complex moral reasoning and cultural learning.
So saying biology made me do it completely ignores like 90% of your biology.
Yeah.
It ignores the biology of attachment and the biology of the prefrontal cortex.
Precisely.
Oh.
A biological disposition toward noticing a novel stimulus does not dictate your relationship structure. Food novelty increases appetite. If you see a new, decadent dessert, your mouth waters and your dopamine spikes.
I mean, mine definitely does.
But that biological reflex does not prove that humans are incapable of repeatedly eating and enjoying familiar, healthy foods, nor does it force you to pick up a fork and eat the dessert. The existence of a novelty response does not render lifelong commitments unnatural, impossible, or biologically invalid.
Okay, if we accept that we are wired to notice novelty, but we also have the capacity for long-term pair bonding, we arrive at the most crucial part of this deep dive.
The practical application.
Exactly. If someone is sitting at home right now, deeply in love, but perhaps in a relationship of 5, 10, or 20 years, and they are feeling a real dip in passion, how do we practically apply this knowledge? How do we use the science of the Coolidge effect to actually help relationships, rather than just diagnosing why they get boring?
The first and most vital practical application of this research is to stop misdiagnosing the problem.
What do you mean by misdiagnosing?
The report highlights how quickly people, and quite frankly, sometimes even therapists, blame the Coolidge effect for sexual boredom. They assume that a decline in desire is solely and inevitably due to habituation to the partner.
Right. We've been together too long. I know everything about you. The novelty is gone. Therefore, the desire is permanently gone. It's a very common, almost fatalistic narrative.
But the data shows it is frequently a false narrative. The report utilizes research by Vowles and Mark from 2020 on a concept called desire discrepancy.
Desire discrepancy, okay.
They point out that declining desire in a long-term relationship is frequently caused by highly fixable, everyday contextual factors. We are talking about chronic stress, sleep deprivation, underlying health conditions, unequal household workloads, unresolved emotional conflict, or simply the sheer exhaustion of parenting.
Right. It is incredibly hard to feel a rush of mesolimbic dopamine when you are actively resentful about who is going to unload the dishwasher, or when you are operating on 4 hours of sleep because the toddler is teething.
Treating every single slump in desire as a biological novelty deficit completely obscures those real, systemic, and highly solvable problems. Before anyone assumes they're suffering from a biological lack of novelty, they must ruthlessly assess the context of the relationship.
Is the engine actually bored, or is it just buried under the stress of daily life?
Exactly.
But let's assume the context is good. The dishwasher is empty, the communication is solid, the kids are asleep, but things still feel, stale. The automatic, effortless spark isn't firing like it did in year one.
That is when we look at true habituation.
Right. And this is where the report delivers what I think is the most critical revelation for long-term couples. It's the stark distinction between partner novelty and experiential novelty.
This is the core practical takeaway that bridges the animal data to human psychology.
It's so important.
The Coolidge effect literature, by definition, focuses on swapping out the partner. But human couples research, specifically the self-expansion research model developed by Muse and colleagues in 2019, shows a completely different, highly effective path.
What did they find?
They found that engaging in novel, growth-oriented activities together promotes significant, measurable increases in sexual desire and relationship satisfaction.
Let me see if I can translate this back to the car analogy. You have a car you've driven for 10 years. You know every squeak of the suspension, every dent on the door. You are entirely habituated to it. Your daily commute to the grocery store in that car is mind-numbingly boring.
Yes, perfect habituation.
Now, you could solve that boredom by trading the car in for a brand new sports car. That is, partner novelty. Or you could take your current car, the one you trust implicitly, and drive it on a completely unprecedented, thrilling, terrifying road trip through the treacherous switchbacks of the Alps.
That is experiential novelty.
Right. The environment, the challenge, the unpredictability, and the shared experience provide the novelty. And your brain's dopamine system, that highlighter pen, still lights up intensely because the context is entirely new, even if the vehicle is familiar.
The neurobiology translates to human psychology in exactly that way. The brain's novelty detectors, including the VTA and the nucleus accumbens, are engaged not exclusively by a new face, but by new information, new challenges, and unpredictability.
So it's not just about the person, it's about the situation.
When you engage in experiential novelty with your partner, you are actively breaking the automatic, habituated scripts your brain relies on.
But what does experiential novelty actually look like in practice? Because I think people hear novelty and they think they need to swing from the chandeliers, completely reinvent their sex lives, or move to Italy for a year.
It does not require extreme measures, no. It requires psychological disruption. The necessary ingredients for experiential novelty are unpredictability, focused attention, and a break from the automatic script.
Give me some examples.
Let me give you three distinct examples. First, travel to a genuinely unfamiliar place where neither of you speaks the language, forcing you to rely on each other to navigate chaos.
Oh, that would definitely cause some unpredictability.
Second, taking a class together to learn a complex new skill, perhaps rock climbing or a difficult foreign language, where you are forced to see your partner struggle and grow as a novice again.
Oh, I like that. Seeing them as a beginner.
Third, engaging in a crisis simulation or an escape room where adrenaline and rapid problem solving are required.
So it's about putting your partner in a new light, a new context, where you genuinely don't automatically know what they're going to do or say next.
Yes. When you are in a highly familiar routine, your brain uses something called predictive coding.
Predictive coding.
It assumes it knows exactly how the evening on the couch will go, so it dials down the attentional resources to save energy. Dopamine stays flat because there is nothing to highlight.
That makes total sense.
By introducing experiential novelty, you force the brain to abandon its predictions. You force it to pay active, heightened attention to your partner in the present moment because the environment demands it. And that focused attention is the absolute prerequisite for renewed desire.
It is so important to note that familiarity is not the enemy here. We don't want to pathologize being comfortable and secure with someone.
Not at all.
Familiarity brings immense benefits. It brings trust, emotional intimacy, security, and a deep, coordinated understanding of each other's needs. The goal isn't to destroy familiarity. It's to realize that long-term satisfaction requires a conscious shift in how you operate the relationship.
It requires a transition from passive reception to active generation.
A shift from spontaneous early-stage novelty to intentional novelty generation. In the first 6 months of a relationship, the novelty is entirely free. The dopamine is free just by looking at them.
It requires no effort.
10 years in, you have to engineer it. You have to actively build a road trip through the Alps.
A decrease in automatic, effortless excitement toward a familiar partner should not be misinterpreted as a loss of profound love. Nor is it evidence that the relationship is doomed by biology.
Right.
It is simply a neurobiological signal that the system has learned the current environment perfectly, and it is time to intentionally generate variation.
So what does this all mean? We have covered a massive amount of ground today.
We really have.
We started with exhausted rodents on a laboratory farm, traced the dopamine pathways deep in the brain, navigated the evolutionary math of reproduction, and ended up discussing how to intentionally engineer unpredictability to save a marriage.
Quite a journey.
If we synthesize this entire journey, the core truth for you listening is this. The Coolidge effect is a real, scientifically validated, neurobiologically grounded phenomenon. It is driven by the ancient mechanisms of habituation and dishabituation highlighted by dopamine. Both men and women possess this novelty-detecting hardware, even if it expresses itself differently based on context and evolutionary pressures.
But the critical caveat, the truth that overrides the biological determinism, is that we are not slaves to those dopamine spikes.
Exactly.
The animal paradigms cannot be simply copy-pasted onto complex human lives. A physiological preference for variety does not equal a behavioral mandate for infidelity. The existence of relationship boredom is not a direct, inevitable translation of rodent biology. We possess executive control, we possess profound attachment systems, and we possess the unique, conscious ability to create our own contexts.
And that leads to a final, slightly provocative thought experiment I want to leave you with. Something to mull over. We know from the science we've discussed that human novelty isn't just about smell like it is for the hamsters. It is deeply tied to complex cognition, to memory, and to subjective meaning.
Which means the very definition of what constitutes a novel stimulus is entirely dependent on your perception and your cognitive framing.
Exactly. So here is the thought experiment for you. Is it possible that your long-term partner, the person you think you know absolutely everything about, whose sentences you finish, is actually an entirely new person every few years?
That is a fascinating way to reframe it.
Because human beings change constantly. Our cells regenerate, our political views shift, our deeply held fears and hopes evolve over decades. If you approach your partner of 10 years with the genuine, focused curiosity that you would normally reserve for a fascinating total stranger, assuming you do not actually know everything they are thinking,
Mm-hmm.
can you effectively hack your own habituation response?
Oh,
Can you trick your brain's predictive coding into realizing they've actually been a novel, evolving stimulus all along, just hiding in plain sight behind a veil of assumption?
it is a profound psychological question. If novelty essentially requires unpredictability and active attention, and humans are inherently complex and constantly evolving, then the assumption of total unbroken familiarity is actually a cognitive illusion created by your own brain's laziness.
Your brain's laziness.
Dropping that illusion and choosing to pay attention again might be the most powerful novelty generator of all.
The illusion of total familiarity. I love that. Thank you so much for taking this deep dive with us today. Keep questioning the assumptions, keep looking under the hood of how your mind actually works, and never stop being curious about the world and the people around you.